Henrik and Howard are just debating here over which one of them, when they stay or go, that we all have to stand up when they do. We'll see who wins on that one. Welcome back. First of all, what a absolutely wonderful beginning of the day. And Mia, from your presentation, Howard to yours, Henrik, yours, and obviously Her Majesty. What a great place to be at KI with this excellent science. If you're wondering, I got the short straw, and I'm the one that's going to moderate the 10 fabulous Goods Prize winners, and somehow try to keep this on time. We will be leaving by 2 in the morning, so just so you all know, which is great. I think, we watched this morning, and really the story of this is Alzheimer's is complex. It's not going to be one shot fits all. The gentleman to my right here over 3 decades ago realized that and said, look, this is ultimately going to be about prevention, combined therapies, precision medicine, and these plaques and tangles. They're the scabs. And of course they're bad, the scabs, right? But we got to understand what's causing the scabs, and it's through understanding what's causing that, along with the scabs, that we're going to find our ways to treatments that I clearly believe my children, my grandchildren are going to be on protocols. My children are going to have multiple options. Henrik, I think that we're not far from a day that when anybody turns about 40 years old, they're getting a blood test, and they won't even realize the blood test is going to tell them, are they at risk, right? And then the protocols and the treatments they go on to. I bring that all up because if you look at these 10 Goods Prize winners here, they're the ones ushering this all in, which is really, really pretty cool. So I want to get us started today. Now, the first thing is we're just going to ask each of you to introduce yourself, in a minute or less, describe the specific work that you're doing in the field right now. And Frank, you were the first Goods Prize winner ever. So, is Nancy in the room with us? Or is she so? Yes. Oh, can we just recognize Nancy Goods? Please stand up. Nancy, how cool is this, right? You need to jump in here for a selfie with everybody, right, on the stage, right? Which is just wonderful, which is good. But Frank, you were Nancy's first, so please.
Well, thank you, Mark, and thank you, Nancy, and your family as well. It's a pleasure to be here, and especially with my colleagues here. So our work is focused on synaptic resilience. So as Howard and others nicely outlined today, there are many factors contributing to dementia, and for us, one of the bottom lines is just the integrity of the synaptic connection, and whether it's tau or amyloid or inflammation, in the end, it's the synaptic integrity is compromised. And we've been fascinated by the observation that there are a very small number of lucky people that even though they accumulate amyloid and tau in their brains, their synapses remain intact and they don't get a dementia. But that's unfortunately a small number of people. So nature is telling us there are signaling mechanisms within neurons that can keep synapses intact. So we're developing 2 drug approaches that modulate receptors on the surface of neurons to counterbalance the degenerative signaling that's triggered by amyloid or tau or inflammation. One of the drugs we've developed has made it through many mouse trials, but into clinical trials as well. We finished 3 clinical trials. And with that drug, what we're seeing in mice is that the synapses are protected against amyloid and tau. So we're excited about that. And now in the human trials, we're seeing a similar finding that the synapses in the humans, in the trial we did with Alzheimer's patients, a phase 2 trial, their synapses appear to be protected. And we did that in part by using biomarkers. In fact, we collaborated with a team here in Sweden to look at the biomarkers in that trial. So we're very excited about that finding. A second drug that we're developing mimics a protein called brain-derived neurotrophic factor, or BDNF, and that's one of the proteins likely involved in exercise and lifestyle effects that we'll hear more about. And just two updates for you. Tau 217, which is one of the blood biomarkers, which is important in the field, again, largely developed by our colleagues here in Sweden. In our phase 2 trial, that goes up in Alzheimer's, and what we see is the first drug I mentioned that we've developed is preventing that Tau 217 to go up. So we're really excited about that biomarker effect. And then finally, we're looking at what's called proteomics in the blood from the patients we've treated, where thousands of proteins can be measured. And the patterns that occur in early in Alzheimer's seem to be counteracted by the drugs we're developing. And so I think there'll be tie-ins because some of those patterns might be affected by lifestyle interventions. And so maybe we're seeing an intersection of drug development and lifestyle intervention. So we're excited to work with our colleagues here in Sweden more about that. So that's the latest.
Literally the shaping of the science. Now, Marty, we already heard from Henrik that usually the first are wrong, but the first. But you were our second Goods Prize winner. So, Marty, please.
Well, to extend the analogy, we're very interested in a pathophysiology process, so the neuroinflammation, synaptic dysfunction. And we initially focused on the neuroinflammation for a particular type, for small inflammatory proteins called cytokines and chemokines. And the drugs that have come out of that, small molecule drugs, which are very safe, orally bioavailable, these were developed purely with biomarkers. So the animal model was driven purely by biomarker, the effects of the drug. And a second shot on gold had to do with specific molecular targets, which Howard mentioned earlier today, a protein kinase that actually is in both the glia, which are the inherent immune cells, and the neuron, which is where the synapses are becoming important. So you have pleiotropy with the same single target in the complementary cells. And more recently, we've worked on one of the primary movers in the neuropsychiatric work that people talked about before is being linked from having the neurons signal to the immune system. Something's going wrong here, and that is a cell surface receptor, and that is in preclinical development. And the idea is to have 3 of these options for precision medicine. All 3 of them have, in 2 cases, they're single molecular targets of pleiotropic action. The other one is actually multiple activities, multiple pathways that's pleiotropic. So it's the concept of being able to have something safe, has exposure in the brain, and it can be taken chronically. In fact, we anticipate many of these will be ones that for prevention, they'll be taken every day, daily at low dose, much like we do with the cardiovascular disease, which Goods actually pioneered in cardiovascular. It's the same exact approach for neuro. And the other one will be episodic, as the clinical demand, especially for the patient and the patient's family, demanded. And to that one, we actually owe motivation for that more recent work to one of our colleagues who's on the panel here, who talked about this need for the family and for the patients and the institution in terms of managing the disease state.
You know, last night I mentioned about how 5 to 10 years ago, these folks were radicals. And if you think about this, the first drugs coming for the plaques, and you hear Marty talking about in the collaboration here, light years ahead of everybody thinking about, but how is this for patients combined in precision? So genius. Now, Nancy, we know we're very proud of, Frank and Marty might have been our first, but our first female Goods Prize winners. And if you kind of look at this panel, there's more girls than boys up here, which is kind of cool. Robbie, our first and wonderful female recipient of the award, please.
Thank you. And thank you, Nancy, and love to Mel, whose photo is there every day when I go to our program of research to regenerate the Alzheimer's brain. And I want to give a little shout out to the mother of one of our audience members, Max Ansbacher, and his champion, his wife, Christine Ansbacher. And it was through Howard's clinical trial several decades ago now that Rowena Ansbacher changed the course of my career and to focus on Alzheimer's disease. And part of that, to come back to what Mark was saying, this innovation row, I would call it, Innovators Row, is the idea that we could regenerate the Alzheimer's brain. We know that atrophy in the Alzheimer's brain occurs very early in this process and is a signature pathology of the disease. And we made a very fortuitous discovery decades ago now, when ADDF was then the Institute for Study of Aging. And I talked with Howard about our discovery, and Howard's advice to me was cut to the chase, Robbie. And we have cut to the chase many times. And because of ADDF's early engagement and support of our innovative idea that we could, in fact, regenerate the Alzheimer's brain, to restore that brain. We completed a early phase 2 clinical trial in which within 3 months we were able to detect in some participants who carry the risk factor for Alzheimer's disease, we were able to detect within 3 months a regenerative response, that their brain was regenerating. And we're now in a phase 2B clinical trial, and using p-tau217 as a biomarker to select participants in the trial who have a high probability of having Alzheimer's disease. So that's where we are in our journey, and we're very excited to report that we're making phenomenal progress using AI strategies for development of a new formulation of aloe that will have the potential of greater use in the clinic once we arrive there. So,
fabulous.
Thank you.
Thank you. Michaela.
Well, it's certainly a pleasure to be here, and I want to thank everyone in this entire field for bringing us to a very exciting moment, I think, where we are making progress. I kind of backed into this field. I'm very interested in episodic memory. How in the world, from just this stream of experience, does your mind kind of make this storage of your experiences? It's who we are, all of your own personal experiences. And, I was very interested in working with some colleagues of mine at Johns Hopkins who were looking at this early phase of transition from normal aging to Alzheimer's disease, and found heightened neural activity in the brain in this condition. And in their first publications thought that was an adjustment the brain was making to compensate so that my memories would work better with aging and greater activation. But I was studying little animal models, and when they got older, if they got higher activity, they had worse memory. And I discovered a treatment that could be given to humans, a very, very low dose of an anti-seizure treatment. And we did this in humans, and the individuals who had heightened memory, heightened activity in their memory-making machinery, and this was supposed to be good for them, when we normalized or brought that heightened memory down, their memories improved. And so now the company I found is working on a whole new class of small molecules that can be orally taken. And they will target a receptor in the brain which is just amazingly loaded up in the memory-making machinery. And it's the alpha-5 GABA receptor, and these are positive allosteric modulators. And we think that they may be just very, very excellent tools for at least this phase of, a very, very early phase of the disease. And that's the other thing I just want to say. The other thing that's going on at Hopkins is that even before mild cognitive impairment, there's something called subjective cognitive impairment. And, that's when individuals, they're still in a normal range, but they say something's changing. I'm having memory problems. And we've now found a biomarker that is positive in those individuals relative to people that are age-matched and they don't have the SCI. And so they're going to be targets, and you want to go as early as possible because you want to prevent the entire thing. And hopefully, you know, when you're getting your physical exam in a decade from now, there'll be biomarkers and people will say, it's time for you to start taking this medication to prevent, you know, some progression that will take you in a bad direction. That's my hope.
Yeah, thank you, Michaela, which is good. Jeff.
So thank you, Mark. Thank you, ADDF. Thank you, Nancy. I'm Jeff Cummings. I'm from the University of Nevada, Las Vegas, and I direct the Center for Transformative Neuroscience. I named it that when I found it because I believe that neuroscience transforms lives when it's done well and develops new drugs. So that's my goal and the goal of our group. Two things that I'd like to emphasize. One is the trial that was funded by the Goods Prize, and that was on rasagiline. It was one of the earliest trials to use biomarkers based on brain imaging to select patients and then to look at the response. We showed that there was a metabolic improvement in patients treated with rasagiline. We were also one of the earliest trials to use tau PET, both as a selection criterion and as an outcome, and we showed a marked relationship between brain metabolism and tau PET. So we're beginning to understand not only biomarkers, but the relationship among biomarkers to understand the complex biology of Alzheimer's disease. Now, more recently, I have developed a passion for following trials in general. I realized that there was great interest, both that our program had and that many other people had, in looking at the progression of the field as revealed by drugs. So we do the annual report. Howard, thank you for showing the figures from the annual report. And we try to understand what's driving the successful trials, what's happening in the trial world in terms of how big they have to be, how long they have to be in terms of achieving the effect that we want. What are the biomarkers that are reporting effectively on drug activity in the central nervous system? So these are the things that the annual report concentrates on. And I'm pleased to say that we now have a new collaboration with ADDF, and Henrik and I were just talking about this, that we would like to do something similar with biomarkers that we've done with the drugs. So I think that drug developers need a resource to go and find what biomarkers would help them most in terms of developing their particular agent. And so we're now starting on a new type of knowledge base that will help investigators access different types of biomarkers to understand how best to show the mechanism of action of their drug. So an exciting next step in what we're doing.
It really is, and what a privilege the partnership is, Jeff. Good. So, Jerry, finally we get back to female winners. So we have a long run of them coming. So, Jerry, start us off.
Thank you. I want to start out by thanking the Lauder family and the Goods family, and of course everyone at ADDF, Howard and Mark. It's an honor to be here. Just briefly, the two programs that I'm focusing on right now in collaboration with these groups. One is focusing on, modifying the trajectory of the disease, through inflammation and neuronal cell death, which Howard did a great job earlier this morning discussing in his presentation. And so what we're trying to do is, there are chemicals in the brain and that are essential for neuronal function, but you overactivate those, and then oftentimes they result in excitotoxicity and death of neurons. And so one of our targets is looking at that, using small molecules to try to fine-tune these interactions and try to suppress that neurotoxicity that develops in the Alzheimer's brain. Another one of our projects is more focused on symptomatic treatments. I know we've talked a lot, and it's extremely crucial to find early detection and prevention and disease-modifying drugs, but there's always going to be that percentage of the population that has well-advanced Alzheimer's disease. And so some of the treatment therapies that we're focusing on are looking at symptomatic therapies. And so one of the drugs or compounds that we're developing is looking at cognition enhancing. And so we've progressed and partnered that program with a large pharmaceutical company, hopefully advancing that into the clinic soon.
Terrific. Mia.
Thank you. I was already introduced, so I will try to be brief, but first, thank you again. Mia Kivipelto, I'm professor in clinical geriatrics here at KI. I'm geriatrician also like Howard, so we early had the interest for the biology of aging, and I'm the director for research and development in theme aging. So Henrik, what you said, connecting the clinic and research, I think is so important, and that's what we have been trying to do. You have an opportunity tomorrow to also see our lab and the fast track memory clinic, and we will talk also about our clinical trials, so this will happen tomorrow. But our research, yes, I have been always interested about prevention. Can you do something before people get so advanced changes or in the brain or in the clinic that you can't do so much about that? And now we know that at least 45% of all dementias are linked to modifiable risk factors, so there is a great prevention potential. However, it has been more difficult to show this in clinical trials. A lot of this earlier evidence came from epidemiological studies. So that's the time when we started the finger trial. And it was the first randomized trial when we took packaged together different lifestyle factors. So I think that was really the secret. It's not enough with single domain. You need to target several risk factors and mechanisms at the same time. Now, as we heard, we have been moving from finger to finger 2.0. We had the worldwide fingers in 68 countries. And I'm so excited about this idea combining lifestyle and different type of pharmacological interventions. I'm very happy for the support from ADDF for the MedFinger trial, which is the first randomized trial when we combine finger and metformin, as we already heard from Howard. And we use the precision prevention idea. We don't give metformin for all. We only give it for persons who have a certain risk profile. And here I think biomarkers will play even more important role in the future. And thinking the next step, I would like to see this as a platform design, adding different arms, finger plus maybe some of the new treatments here. Semaglutide was mentioned, that's something we are discussing now. Of course, anti-amyloid treatments and so on. So precision medicine and next generation of clinical trials with the partnership of ADDF is my hope for the future.
Love it. So keeping the speed going here with Innovators Row, Miranda.
Thank you so much, Mark, and Nancy and the Goods family and ADDF and Mia for hosting us. This is fantastic. So my lab is really interested in the greatest risk factor for developing Alzheimer's disease, which is aging. And so we heard a lot about the biology of aging. So this is a field of science that has identified fundamental factors that happen in our cells and tissues as we get older, and they're really conserved across species. So the same things are happening in worms and mice, in dogs, in humans. And once we've understood these fundamental factors, then the question becomes, can we do something about it? And in the laboratory, one of the hallmarks of aging is called cellular senescence, and Howard introduced that a little bit. And so what these cells are, these are cells that we all accumulate in all of our tissues as we get older. And this happens through the course of our lifespan. And these cells figure out clever ways to survive. So they really should be dying, but they don't, and they stick around. And when they do stick around, they become very toxic to their neighbors. So they release a bunch of inflammatory molecules. So they get to live, but they kill everybody around them. And we, in the general biology of aging field, have determined that if you clear these cells throughout the lifespan, you can extend the healthy life of an animal. You can actually extend the number of years or months of life. And so we identified that these cells are accumulating in brains of Alzheimer's mouse models, And when we remove them with drugs, one is a repurposed chemotherapy combined with a natural compound. When we treated mice with these drugs, we improved their brain structure and function. And these were mice with advanced disease. So the ADDF has funded our clinical trial. We completed phase 1 that was safety and some early positive biomarkers, really too small to get too excited, but the ADDF is funding our phase 2 trial, and we are currently launching our study in Spain.
Fabulous. And just a point there with ADDF and when the organization started, Howard really saw us funding the Valley of Death, and that was how do you take these great ideas that are on the bench and get them into humans? And you're hearing that here. You're also hearing that there's a second Valley of Death when you get to this phase 2 to get powered enough for phase 3. And that's kind of the cusp that we're on now, but we are here for you, absolutely here for you. Rhoda, is like digital going to be the new blood or what? Sorry. Rhoda, can you please tell us about your work?
Well, first of all, I'm very humbled and grateful to the Lauder family, to the ADDF. I'm so honored to be a Goods Prize winner, Nancy, and to Mel as well. And I wanted to start out by saying, it's funny, that line between innovative versus crazy is a very small one, right? And so it's interesting where I sit because digital is so new. So that's the realm in which I like to think about. And for a very long time now, it's been more on the crazy side than on the innovative side. So to be in front of you today is really, really a tremendous, I think, show of how much the Diagnostic Accelerator was willing to take a chance, much in the way that you've done with many of these others and with the blood-based biomarker. But digital is still in its infancy. And this concept of digital biomarkers is the one that I'm really trying to think about. So we talk about the importance of earlier. But what do we mean by earlier? So right now, I think about it from a behavioral standpoint. Because to have the pathology of the disease we know is not sufficient. And so we've talked about the probability that you're going to come up with Alzheimer's on the basis of these biomarkers. What I want to focus on is who is that person going to be? And how are we going to pick it up? And how are we going to pick it up so early that we can actually prevent it rather than just focus on treating it. So that's really the realm of digital. And the other thing that I think a lot about is how do we bring this science to anybody, anywhere? So when we start to combine the concept of blood, which people know how to collect, they know how to store it, and then they know how to use it for these discoveries. Digital actually has the same qualities. But because of the smartphone, right, that's a digital data collection device. It has multiple sensors in it. And they can pick up information about how you're engaging with the world. And at the end of the day, everything we do through our brain. So as we're engaging with this smartphone, we're actually emitting our cognitive capabilities. And now, if we can do this on an ongoing basis, we can start to detect much earlier on. That time, what you call subjective cognitive decline, we can now call it objective cognitive decline. And I think we're going to redefine what we mean by asymptomatic. I think that we're going to move that boundary even further. And symptomatic will be within those what we call normal ranges, and then your interventions of modifiable lifestyle risk factors combined with some of these early drugs. I think that's what's going to lead us to the future that lies ahead.
And just to prove she's not crazy, show of hands, how many people tracked their sleep last night? There we go. We're on the way. Henrik, I think it was Frank that described coming here to Sweden and being with at KI, being with you and in Gothenburg, that this is the capital of biomarkers and the explosion in biomarkers. So, Henrik, I know we've heard a little bit, but just as a Goods Prize winner, please.
First of all, I would never track my sleep, but I try to do Mia's 5 fingers now. But it's not completely easy. Can I tell a little bit of the story, Mia? A few weeks ago, Mia and I, together with the Swedish Alzheimer Foundation, were at a big celebration with Swedish stars, people singing, and musicians, artists. And then Mia talked about her 5 fingers to prevent neurodegenerative disease changes and dementia. And then afterwards, I was invited to an after party with the choir.
Yeah.
Hellmans Drängar. Me and my sister went there. They know how to party, that I can tell you. But then there was a discussion, and I was asked about this as an expert, sort of, about your 5 fingers, because one of the singers said, "Here I am." He was a little bit chubby. I sing and I drink beer, thereby socializing. So that's 2 fingers. But then some, no, that's just 1 finger. One of his colleagues said. So I think I have 3.5 or 4 personally. But now I have recruited Rhoda's son. Yesterday I met Rhoda's son. He won't work with digital biomarkers. He will work with molecular biomarkers. And then we will collaborate. So from my perspective, the molecular biomarkers that I talked about, they tell about tissue changes in the brain. But to me, it's extremely important to combine that information with brain function data. So we have started to collaborate carefully, Rhoda and I, after this animosity, to see if we could make something together. And then you will have the smartphones, and we are trying to develop a finger prick test for neurodegenerative dementias. And that's also thanks to these new discoveries that have been made in regards to making the tests even more sensitive. So now we can measure amyloid, phosphorylated tau, neurodegeneration, astrocytic activation, and some preliminary data on microglial activation in 1 drop of blood. And now I hesitate because it sounds like this fake company, Theranos. So yeah, but this is sort of real. It's reproducible actually. So that's one of the things we're working on. Yeah. And then we are trying also to address one key pathology, as I mentioned, TDP-43. See if we could get better fluid-based biomarkers for that pathology. Also collaborating with some researchers working on imaging. And then a final thing is actually a synaptic health index, looking at some of the pre-synaptic markers that behave a little bit differently in neurodegeneration. There is a very effective ratio if you take SNAP25 concentration in CSF and divide it by the NPTX2 concentration, then you get, it seems like if you're normal for that ratio, and have amyloid, you will not decline. So that could be an interesting collaboration also in regards to some of the approaches mentioned here. And I would also, again, like to thank the Goods family, the Lauder family, and ADDF, of course, for supporting this and bringing all of us together.
So, Henrik, I have one more person to add to your collaboration, which is Frank's son, who was there with us last night. And I'm not sure if he's here today, but he's here for the proceedings. He's going into AI. And so, we're going to add him to this posse. So the ADDF at its origins in 26 years has always been about 3 things, and you've heard it all here beautifully today, and the Goods Prize Award just represents it. Biomarkers, prevention, and treatments, and I think those in the science world know this, but for others here, the collaboration between those 3 pieces to get to the patient. So that brings us to Howard. And Howard, so my question is, who's your favorite? Don't worry, I won't do that to you, Howard. Actually, we're going to start with Howard, and we're going to go down the line. We've talked about this, that there are these different pathways to aging, and there are biomarkers associated with the pathways. So, Howard, when you think about in the presentation that you did today, what are your two which you think are most promising pathways as we sit here today?
Yeah, I think inflammation and metabolism, insulin resistance are really the low-hanging fruit right now, because we really understand inflammation pretty well at this point, especially the unique features of inflammation in the brain and in the aging brain, and there are therapeutic targets. So, and they're in clinical trials, and so for all those reasons, I think inflammation is going to be an important adjunct to, for example, removing the amyloid with the current disease-modifying drugs. And insulin resistance, I mean, metformin, semaglutide, these kinds of drugs. I think because of repurposing especially, we can really ask the question in human beings and know the answer in 2 or 3 years, and we'll have answers, at least with the drugs that are being tried, and that can have a big impact.
Yeah, inflammaging, as I learned today, right? Which is good. So quick down the road, Henrik, same question?
Yeah, I am an amyloid guy. Sorry.
This is great.
I really, but I really think also it's getting more and more clear that we react differently to that pathology. But I'm a little bit traditional here, I would say, but I think it's very clear that if you have amyloid in your brain when you're 70, it's hard to become 80 without cognitive symptoms.
So let me push you on that. What along with amyloid?
Yeah, then we come with what you said, actually. So it's all about this tissue reaction. So it really seems like when amyloid plaques form, there is an almost immediate tissue reaction to this pathology. And that is reflected in the biomarkers in a very strong way. I think that's why it's been easier to get good biomarkers for Alzheimer's disease compared with frontotemporal dementia and Parkinson's disease. Even though you have neurodegeneration, we have a hard time measuring it.
So just along those lines, we know from the clinical trials with the anti-amyloid that you can completely get rid of the plaques, and it slows it down by 35%. And the EMA is asking whether that's the European agencies and even the FDA. People are asking, even clinicians are asking, is this clinically significant? So there is an effect there. It's associated with side effects, but there's an effect there. People are going to go on these drugs. But the question is, how do we get to the other 65%?
Yeah, but that's really important, and that's why I think it's so. I mean, all the things we have discussed today will be really, really important.
So, Rhoda, how do we get the other 65% from your view?
Yeah, I mean, I'm a little bit agnostic to actual pathways, right? And I think that we all know that it's multi-etiological, right? And it's probably the fact that it's multi-etiological in a dynamic way across your lifespan. So where you start with your treatments and where you end are probably going to be different because other comorbidities are going to come in, right, and create new pathways. So I think that from my end, from a digital standpoint, the challenge then is to make sure that we have measures of those clinically meaningful shifts that are happening. Either that you're now positively reacting to whatever drug you're on, when you start to plateau, when it stops to be effective, and where do you need to pick up and move in a different direction. So I don't think of anything as static. I think of it as constantly fluctuating. And then what do I do in terms of thinking about these digital biomarkers, and how are they going to then give us insight to those changes as they're occurring? So I don't think of it as purely biological, right? I mean, I understand that there's a biological underpinning, but I also think that, no one goes to a physician and says, I'm feeling an increase in amyloid, right, or tau. What they're saying is, I'm having problems with my memory, right? I can't execute on the things that I have before. And those are the ones that are meaningful, and we got to detect it much, much earlier.
Well, I'm not sure, because with, Henrik's finger prick test, maybe they will be saying, I'm feeling a lot of amyloid today.
Yeah,
just kidding.
right.
So, Miranda, Synalytic's point of view, but also a point of view of, you came up in the science when this whole thing, you never were bought into a debate whether it's amyloid or not amyloid. You had a clearer kind of perspective on this, so please.
So actually, as a graduate student, it was definitely still, tau was still on the cusp. It was very amyloid focused. So biology of aging was definitely out there. So thanks to the ADDF and everybody that, has really paved the way, it was far less difficult for me to to convince people that targeting biology of aging was important. And it's really interesting because biology of aging and how we age, as Rhoda just said, is not linear. And there was a beautiful paper that came out just a couple of weeks ago showing that there's two major shifts in our life. And this was done by continuous monitoring of blood and hundreds of thousands of metabolites in blood and urine and breath. One of those shifts happens around the age of 44. And so, when we think about aging, we often, I think historically, have studied aged, not the aging process. And so we're really interested in understanding when in our adult lifespan do senescent cells first appear, and looking at post-mortem human brains from people that passed away age 20 all the way to 100 plus. And this is really, spearheaded and funded by the Goods Prize. We're finding senescent cells in people's brains in late 30s, early 40s. So this is, if we could find biomarkers to be able to detect that in the blood, we would be able to know, you may have a senescent cell burden, clear these senescent cells and come back in 6 months, 9 months, a year, who knows how long. But because senescence as a hallmark of aging really ties into so many other things like inflammation, like Howard said, metabolism, all of these things. And one of the areas that I'm really interested in and really pleased that there's so much research effort is on the immune system and how the immune system is really impacting the brain.
Fabulous. So, Mia, from a pure lifestyle perspective, what two other areas combined with lifestyle do you think about?
So much. I mean, I come again from the precision medicine combination therapy idea. I don't think there will be one magic pill solving the whole thing. It's like the cancer, HIV, cardiovascular diseases. It's the combination therapies. And I think, of course, I mentioned about the Medfinger trial, where we are now moving to that direction, adding different, starting with the repurposed drugs because they are more widely available. I think semaglutide is very exciting because of the inflammation, glucose, metabolic things, and that makes sense to add that to the platform. That's, I think really the next candidate for us. And then, as I already mentioned, taking all the different approaches from here. But we are already also thinking kind of triple therapy. Why not thinking the anti-amyloid, there is effect there, lifestyle, there is nutrition-based interventions, starting building this package where you can really have different type of mechanisms and get even more sustainable effect and possibly even lowering the side effects if we take care of other factors and comorbidities as well. So I think designing the study design for that, how we can do it, will be very, very exciting. And here I believe both the blood-based biomarkers and digital biomarkers will be very important.
Jerry?
I tend to agree with Howard on most things, but so I also agree as far as pathways are concerned that inflammation is a really hot topic right now. I think one of the things that I am most excited about is how, as a research community as a whole, we're not hyper-focused on any one pathology or any one target. As you've heard numerous times by several individuals, I don't think any of us believe that there is a magic bullet or that there is one cause of Alzheimer's disease or one cure. And I think it's really well represented by the panel, which I know is very intentional on Howard and the ADDF's part, in that we focus on many different aspects of the disease. If you're looking at, prevention, modifying the trajectory, early detection, symptomologies. And I think we need to really hyper-focus on individualized medicine. And so looking at the individual, assessing what stage they are in their disease, what their individual underlying pathologies are, what their symptomologies are, and then developing a treatment plan that's tailored, for that patient. And I think that's where we're going to make our huge advances, in trying to cure this devastating disease.
Jeff, anybody you violently disagree with?
Not exactly disagree, but I bring a different perspective to this, which is I'm really excited about success. So we went from 2004 to 2021 with no new drug approvals for Alzheimer's disease. Almost 20 years without a single approval. Now, in the U.S., 2021, 2023, 2024, new drug approved in the United States. They have one thing in common. They remove amyloid plaque from the brain. Very powerful drugs. So is it important that it's amyloid? Not particularly. It's important that science has shown that this is a disease with vulnerabilities. And we've had one breach in the wall that separates us from success. And now we're going to have more successes. And I agree with Howard that there's 30% slowing. That's great, but it's not enough, right? It's not enough. So that leaves us with the 70% that we need to solve. And that might be combination therapy, or maybe we can do better with the antibodies. And I think that's another opportunity, is can we get more of them into the brain? We can see really clever technologies evolving for that. Can we stop some of the side effects? That's like a separate treatable problem. So I think the antibodies themselves have shown so many things that I'm excited about. And they've also opened up a bunch of new questions and opportunities that now have to be addressed.
McKellar, is he crazy or is he?
No, he's on to something. I'll keep listening.
That's right.
I'm really interested in individual differences and variability, biological variability as well. And so a lot of my basic research has been with outbred animals that do age differentially. And so there is something that's called, successful aging, that even as you get older, you maintain good functional performance. And some of the most interesting work that I've been working on is that those animals in this model with biological diversity that age well are not in their brains like young adults, that they actually have adaptations. So if you think, we think about plasticity as something that happens during development, gargantuanly. But it also can happen later in life, gargantuanly, I think. And I think that just sort of understanding more about what the opportunities are for healthy brains, adapting to circumstances so that we have better outcomes is a possibility.
So Robbie, progression, resilience, does that ring any bells for you?
Yes, and so something that we have focused for many years on is what I now term transition state biology. Right, so many people in the room who've had an experience of a loved one developing Alzheimer's disease, oftentimes you'll hear, Mom was okay until, Dad was okay until. And what we have focused on is a midlife transition in the woman's life of aging, the menopausal transition. And what we have discovered is that, yes, this is reproductive aging, but it's brain aging. And that's when this vulnerability can emerge in midlife, right? That this transition state is not unlike puberty, right? So in puberty, these risk factors for this risk for depressive disorder or schizophrenic disorder can emerge, right? And it has nothing to do with reproductive capability. And it has everything to do with this transition state or this, this renovation project on the brain. And so what we have discovered is that for women who are at twofold greater lifetime risk of developing Alzheimer's disease, it's not because women live longer, on average about 4 years. It's because the disease can start earlier. Now imagine, right, we come back to what is the biomarker, right? What is the biomarker that can tell us that that woman now is at risk for developing Alzheimer's disease later in life? It turns out inflammation is a major outcome, can be a major outcome in this menopausal transition. Metabolic dysfunction, glucose dysregulation. So now all of a sudden, We can intervene in midlife. We can now understand and have a fairly good prediction about who is at risk, and now intervene in midlife to put that brain on a successful aging trajectory instead of a vulnerable one.
Fascinating. Absolutely fascinating. Marty?
Well, I have a vested self-interest in inflammation, yes, but I mean, my bigger picture thing is age is just a number. And this is yesterday's technology. And with things like today's nanomedicine, you will be able to take a very safe, biologically compatible pill that will report back, so you won't have to prick a finger. You'll be able to do inflammation markers. You'll be able to use neuromarkers. In terms of the other part of nanomedicine is nanobodies, which basically are taking the promising therapeutics and they're getting into the brain with less side effects. But you can also put them on chips that the average person who has a smartphone would get a kit, just like a urinalysis kit that you might test at home, or a pregnancy test you might, but you would actually have something in which you would have the specific thing on that kit, and you would actually plug it into your smartphone, and it would tell you, it would record it, and it would turn it back. So you're going to have personalized medicine as individualized medicine because you have a profile. So you'll know at whatever age they're at that you have a propensity for intervention, because maybe you don't want to intervene with certain anti-inflammatories if the person is a high-intensity athlete, where that brings down the target's levels. So basically, they have a non-pharmacological treatment that they're in process, but as they age, maybe they'll can't exercise as much, you'll have to intervene, okay? We have this with amlodipine in cardiovascular. It's the world's widest used drug. It's very cheap. It's, so it will evolve that way. So I think this is the way it's going to be in the next 5 years, not 10 years or 20 years.
Yeah, I love it. And we're going to come back to that. And you see, Rhoda, Marty's as crazy as you are. So we just got Frank.
Yeah, I'm excited about just the omics, and that's how that's going to accelerate us. We saw back in the 90s how DNA sequencing just revolutionized parts of medicine, right? The genetic disorders. And I think now, in 2024, with the layers of omics that will become feasible at a reasonable cost on individuals, and I'm dealing with proteomics now, but metabolomics. So the individual biomarkers in parallel with proteomics. What are the proteomics telling us now? That Alzheimer's is unfortunately a very powerful biologic disease. I call it deep biology. This is not something just at the surface. It's as powerful as a terrible cancer, unfortunately. And just going after one target, just like in most cancers, won't be enough, unfortunately. So we need these pleiotropic approaches. And the omics are telling us they're already abnormal before symptoms start. So the omics will tell us things over time. The omics are already telling us that people from different backgrounds have different patterns, right? So the precision medicine will be a 10x amplification with the omics that are becoming available today. So I'm imagining a highly customized, omics-based, pre-symptomatic, pleiotropic, prevention or therapeutics or both, some combination of lifestyle and heavy, intense drugs. I like the pleiotropic drugs. Metformin was mentioned, but rapamycin or some of these other drugs that affect multiple mechanisms. I think the field will continue to accelerate with that kind of technology support.
It's absolutely fabulous. I had some follow-on questions from the group, but as you've gone, you've all answered them, including the last two, which is, where does this look like in 5 years? We're going to open this up. There's 2 mics that people come up to and ask questions, and I'm going to reserve the right to ask the first 2 questions. So if you have questions, please come up to the mics now. I promise I'm going to get to you. I have one for Mia, and then I have one for you, Howard. Mia, because I hear this all the time from so many people. Right, of all these prevention measures, of the 5 fingers, what are the one or two that are most important?
That is exactly, that's a very, very good question. Activate all 5 every day. You don't need to do so much like Henrik mentioned.
But the
do
one or two,
fine.
Mia.
One or two.
Yeah.
It's individualized. Maybe something is good for me and something for you. So here I really believe we come back to the individualized, personalized medicine, knowing what is my most important risk factors. That I think is the future. I also like what you said, Robbie, starting early. That's also the thing. It's never too early to start to prevent. And now we are moving to even younger ages. We have the initiative High 5 for Life, when you can start shaping your life when you are even younger. That's the direction.
Right. Howard, I want to come back to something that Frank said. He talked about the field of cancer. And you look at the field of Alzheimer's 40 years ago, where it is today. How does that compare with cancer, and what can we learn from cancer?
Yeah, there's a great book called The Emperor of All Maladies, and it describes how cancer research evolved from like the late 1890s, when X-rays were first invented, and it was sort of a biomarker for cancer if you had a solid tumor, and then started radiating people, and then surgery, and then Farber came along, and he's just started throwing drugs at people to see what would happen if their cancers would shrink using, looking at it on X-rays and so on. It's a really interesting book, and I think in some ways Alzheimer's research is paralleling that, but we're in a very strict regulatory environment, which we weren't in when cancer treatments were evolved. So even today, you have cancer patients on 4 different drugs, and these are considered protocols. And the reason is the cancer cell has gone out of whack in so many different ways that it's like whack-a-mole. You have to treat the multiple pathways where the cancer cell is gone out of whack to really conquer the cancer. And the difference is that in the brain, neurons don't divide. Cancer cells divide, they proliferate, and they form tumors, and the tumors in themselves are what kills people, right? So you can measure that and try to prevent them from proliferating. Even though the pathways are similar in the brain, you need to, the neuron is dying from many different mechanisms. And so it's very similar to cancer. It's why we can repurpose cancer drugs for the brain, because the pathways are similar. It's just the outcomes are different because neurons are not proliferating. You lay down those memories and you want to try to keep them for life. So you need those circuits. So I think we can learn a lot. And one of the things that I think is very timely now that we're going to learn a lot from cancer is combination therapy. Because that's where it was discovered that you can give a cancer patient one drug, doesn't work so well. You give them 2, works a little better. You end up giving them 4. And so the cancer field has really had to innovate on how to do combination therapies. And we recently had an advisory board on combination therapy with experts from around the world. And one of the experts was a cancer specialist who taught us quite a lot, I think, and is going to help us quite a lot in figuring out our strategy. The other difference is that in the era of cancer development, where you had multiple combinations of treatments, things weren't quite as expensive. You could kind of do it. But in this era, those kind of trials are going to be much more expensive for combination therapy for Alzheimer's disease. And I think we're going to face more challenges in developing combination therapies, but it's definitely going to be the way to go. And we could have a whole day of discussion about this issue.
Completely on that. And just a note on that group that was put together, all the 4 major pharma players that are in this space were all part of that group, and there's going to be a report out for them a little later this year. All right. Jacques, do you have our first question, please?
First of all, thanks for ADDF. But I have one question. Combination treatment is mandatory. Precision medicine is expected using biomarkers. But I think in the years to come, one of the most difficult problems to resolve is how can we organize innovative clinical trial design, because it's a very new world.
Rhoda, did you want? Or Jeff?
Well,
Go.
right,
And
I'll go.
I'll talk about this a little bit this afternoon too, but you're absolutely right. We have a plethora of targets. We have preliminary information on many of them. So how would we prioritize the combinations? Because we can't do them all. And I think we need a lot of ideas, and we welcome your ideas, Jacques, about this. But one idea is to use more computational strategies. And I think the computational tools are growing sufficiently well that increasingly we can use omics as well as genetics and many other types of information to begin constructing models that would allow us to theoretically test drugs and then choose the drugs that could be put into the clinic. So I personally am excited about the promise of computational models to help us with the prioritization problem that we have.
Yeah, okay. Rhoda, then Mia, then Howard.
So I don't do clinical trials. And so I'm not weighed down by the pragmatics of that, right? So I'm allowed to let my mind wander. I think that the future is definitely virtual clinical trials. And to do virtual clinical trials means we're going to have to revamp the entire way in which we think about this. But now, if we're monitoring, and the other thing that we'll be able to do with virtual clinical trials is be far more inclusive, because that's been a real barrier to actually the effectiveness. We talk about precision medicine. We're not going to get to precision medicine unless we find ways to do this much more inclusively. So the future is, to me, putting it, not having people come to you, you've got to go to them, and you've got to make it work. And now think about the fact that if you have these blood-based biomarkers, you have your drug treatments, whatever combination, and you're monitoring for the clinical effectiveness of these. And I think that that's where the future is, but we're going to have to rethink how we do clinical trials to get to that reality.
Mia.
Fully agree. I think exactly if we move, and we need to move to the combination therapies, there are many regulatory, many operational strategy questions, and having a broad collaboration, early discussions with FDA and others, I think is very important, how we shape the future of clinical trials. And learning from other fields like cancer, how they have been doing the combination therapies, HIV, even COVID. So I think there is a lot we can learn. I mentioned the platform design that could be a starting point for this kind of thinking. You start with lifestyle, you add different arms, and the precision medicine part is there, that you match the treatment with the underlying pathology. But it needs broad collaboration. No one can do it really alone. And Rod, I really think what you said is there. We need to rethink also when we start. We want to start even earlier, and I agree. Then maybe the persons are not coming to the clinic. We need to do the trials in a more broader way, and diversity, it will be so important for the future success.
Right, Henrik Howard.
Just a quick comment also that if you work on also people who might be interested in developing drugs and examining certain pathophysiological pathways, make sure to think about the drug biomarker pipeline, a biomarker pipeline that parallels the drug development. So that one also in the preclinical stage, don't throw away cell media. Keep it. Of course, you have to throw away a little bit because otherwise your freezers will be overfull. But in the cell media and in the biofluids from your animal models, there you could do explorative work and find the biomarker signature in an accessible biofluid for what the drug is expected to do. I think that is an important supportive action for what we discussed now.
Fabulous, Howard.
I just wanted to comment. Frank has brought up omics, and we've used the word, and I'm not sure the audience really knows what omics is. But I mean, I could briefly maybe comment. Frank, you probably can make a better comment than I can on that.
Yeah, I mean, in terms of omics, say proteomics, right? So the ability to measure, 1,000, in the order of 8 or 10,000 proteins all at the same time. And studies are doing this now in blood, CSF, and then human brain postmortem. So 3 layers, right? And what's emerging are what they call, modules. So a group of proteins that might be indicative of a synaptic state or inflammatory state or autophagy state, right? So we're seeing human biology laid out before us in a way we've never seen before. And I think that'll help with trials on both ends. Number one, what's your omics pattern? And you'll declare yourself as a subgroup. And maybe that's the subgroup. We'll try something. And then we'll get faster readouts, because maybe within 3 or 6 months of this therapy, whatever it is, we'll know so much more about, did it engage the biology or not? It used to take us forever to know that. So I think many aspects of omics accelerating us.
It's a good example of how technology really advances the field, because it's new technology, and you go from measuring one protein at a time to thousands of proteins, and then you can see patterns.
Right.
It's a big advance.
Right. But I think it's a kind of a sister technology to the individual biomarkers, because the individual biomarkers we can still interpret better, right? Like the ones that Hendrik and others are developing. So I think they'll have to go together, because when you measure 10,000, I don't think you're as informed, necessarily. They complement each other.
Marty, please.
Jumping back to the question about combination trials, informatique is the foundation of AI. That the actual expression is garbage in, garbage out. So informatique takes care of the garbage, basically, if you've curated your information. Combination right now, using AI, the International Commission on Harmonization has more recently pretty much defined what the risks are for drug-drug interaction on any individual drug coming forward. So basically, in terms of building a database, immediately you start at the end, grab all the drugs that are approved, grab all the drugs that are under trial, and go into deep dive on that informatique and ask, there's no sense trying this combination because we're going to have toxicology. So virtually, we know there's going to be toxicology. Don't touch it for now. Put it on the shelf. Okay. Then you bring that subset in, and then you go to the research literature and pull in your omics, DNA, genomic sequencing, DNA sequencing, protein sequencing, and you pull it together. And actually, that gives you a tertiary organization. But actually, starting at the end and starting tomorrow, there's great hope there, I think, just from the information it's based on.
You're fascinating. Absolutely fascinating. Please, next question.
Hello. My name is Maria Eriksdotter. I'm professor at KI. I actually had the same question that you already answered. I also think we're going to have a lot of combination therapies, but maybe 3 or 4 drugs, prevention and other things, and how you design that is really going to be the key question. So, but you answered that, very well, I think. So I just have one more question, and that is, what is the impact, do you think, about sex differences? And do you see that a lot in your work?
So let's start with Robbie. Great question.
So yes, this won't become as a surprise to human beings and the scientific community. We age differently. There are different stressors. And this midlife transition in the female is one of those midlife aging transitions in which vulnerability can emerge. The same holds true, interestingly, in men who are given prostate cancer therapies. So interestingly, that same constellation of symptoms can emerge that we typically associate with menopause can emerge in men who are on prostate cancer therapy, suggesting that there are systems of biology that are comparable under these two kind of reproductive or endocrine aging events. Differently, what we find, interestingly, is that there's a huge difference between the inflammatory profile between the aging female brain and the aging male brain. Interestingly, and we also find another interesting sex difference, that when we look at the metabolomic profile of ApoE44 males, metabolically look like an ApoE33 female. So there are sex differences, and then there are these avenues, these opportunities where those sex differences can actually inform how we understand this aging process in females and males, and therapeutic interventions that what we think would be targeted to the female, but in an ApoE4 male may in fact be quite relevant, that we typically associated with the female. So there are many sex differences, as I'm sure you well know, and when we look at the precision medicine, precision approach to therapeutic prevention, precision prevention, and precision treatment, we are going to have that better understanding of what sex, chromosomal sex, brings to that therapeutic portfolio option.
Yes, this is a great question. Actually, tomorrow we will have a small workshop titled Sex and Gender Differences in Alzheimer's Disease, the Gateway to the Precision Medicine. So I really believe here we can learn so much more about that. I would also say in many studies, the sample size has been the limitation. It's difficult to really study the sex differences. Now in the Worldwide Fingers, we have an international working group where we aim to collect data from all countries, all cultures. We both have the sex and gender aspect there, and I think that may help in the future to get more answers.
Fabulous. Next question.
Thanks. Hi, Jose Luxner from Columbia University in New York. Thanks for all the panelists, and congratulations, Henrik, on your award. So I have a very practical question, which is, now that donanemab and lecanemab are approved in the United States, if we do clinical trials in people with MCI and early dementia, are we obligated to do a biomarker and offer potential treatments on all those participants? For example, I have a clinical trial of metformin where we don't do biomarkers. And so if we go to a phase 3, I'm wondering, will I need to permit, the use of lecanemab or donanemab in participants, although there's barriers to access, but if that weren't a problem, and what's ADDF's approach to this when you review, grants to ADDF, for example?
Good question. Howard.
Well, I think our group is still thinking this through, and it's just an issue that's starting to come up. I mean, the way I think about it is that it's what we call a 4-factorial type of design, right? So usually you have a 2-factorial design, a placebo, and the intervention. Excuse me. In a 4-factorial design, you'd have the one drug by itself, then the other drug by itself, the 2 drugs in combination, and the placebo. I think I got that right. And there might be other variations. And so when you go from a 2-factorial design to a 4-factorial design, you're increasing the number of patients you need and the cost of the trials enormously. So I don't think we've really come up with an answer to that, to be honest. It's one way or another. I mean, obviously, in the monoclonal trials, everybody has to have amyloid, and I think for people that we're trying to test for Alzheimer's disease, we want to know that they have amyloid just to know that they have the right diagnosis. And we could have people that, some of the other variations are people that are on the monoclonals, and then they go afterwards, after their treatment, say 6 months on the monoclonals, the amyloid is removed from the brain, as you see on PET scan, and then you put them on the second drug, like metformin, to see what happens. Or do you start them both at the same time in the beginning? So a lot of questions that the field hasn't answered yet. These are all really important questions right now.
Henrik.
Jose, I have the answer. Do your study in Europe.
That's right.
There we allow for this type of almost unethical study design.
Okay, all right. I'll be coming to Stockholm a lot.
I'm going to do something that's going to tack towards a wrap-up, and I'm only doing that to buy you all time, because I'm going to ask a rapid-fire question. We're going to start with Frank. We're going to end with you, Howard, so you get last word as always. And I'm going to tell you the question now so you can think about it. Then I'm going to do a little bit of a wrap-up. Then I'm coming to you. It's January 1st, 2030. A new decade is starting. what's the biggest advance that you see that's going to happen between now and then? And just turning towards a wrap up, and we've heard a lot about the Lauders and the Goods family and the ADDF and 26 years of doing this. We at ADDF we began as ADDF 1.0, and that was very much a preclinical phase, and it was this phase that we were just exploring everything. And 2.0, the phase, which was more like around 2016 or so, or 2015, came the time of pharma was going after amyloid, right? What's with that scab? And so the view was, Howard's view was, went to the board, we don't need to be pursuing amyloid anymore. We exist to get this into the hands of pharma who can take it across the finish line. So the pivot at that point was this complete biology of aging. What were all these other pathways? We started doing these multiple shots on goal. As we go into the new year, and it comes off of the last question, you're going to hear about ADDF 3.0. And not to worry, we're still going to keep taking these multiple shots on goal across all the pathways, but we are pivoting very clearly now. We're going to put capital behind the beginning of combination therapies, the further push on prevention, the biomarkers needed to really get precise about the combination therapies, and the precision medicine. And that's what you'll see going out to the end of the decade, ADDF 3.0. So here we are. It's the end of the decade, Frank. It's January 1st, 2031. Got to be quick.
Yeah, it's the end of the decade, and the big difference will be the intensity of biomarker information at low cost that we'll have for everybody. And every trial that's done will also be measuring the biomarkers. So with all the potential therapeutics, they'll have a signature, and each of us will have a signature, and we'll have opportunity to match.
I love it. Marty?
Again, age is just a number. So following up on that, you will have the profile that you have, and whether you're 30, 40, 60, or 80, the decision will be made based on your profile.
Interesting. Robbie?
So perhaps just a little bit further out, I actually vision there won't be a diagnosis of Alzheimer's disease. You'll receive a diagnosis of you are at risk for Alzheimer's disease, and here is a therapeutic strategy for you, precision therapy that will put you on a trajectory of brain health, sustained brain health.
Phenomenal. Michaela?
Yeah, so one of the things that I think, and this is very much to do with biomarkers, is having things parameterized and normalized. I don't know what a biomarker or a change value that gives you a meaning for where you are in a biomarker space relative to disease. And, I think that's a very fuzzy area because it takes a huge amount of data to arrive at that kind of understanding of what your parametric space is. And, maybe people will differ on that approach, but I think we need a lot more information so we know where we are in the biomarker space for individuals.
Jeff, January 1st, 2030.
Okay. We'll have biomarkers for the pathology that is present in the brain. We will have treatments for many of those pathologies in the brain, and we'll actually be treating people for the conditions they have.
Fabulous. Jerry?
You're going to hear this a lot, I think, from us, the theme of biomarkers, but I think to expand on the previous panel members, the use of biomarkers to not only do patient selection for trials, but also your primary outcomes. And I think that because of the use of those, that you'll see a significant increase in the success rate of clinical trials relative to our, 99% failure rate right now. So,
Mia?
I hope it will be the decade of brain health. And the brain health checks early in life, knowing exactly what pathology you have, biomarkers, and then having the tailored, personalized interventions. And the second thing, the implementation gap. I hope we have been preaching that so that we have blood-based biomarkers in use. We have the anti-amyloid therapies also in Europe. We have the first platform designs up and running, and we can also share data more easily that we can speed up that process.
Miranda.
What I hope is that we have biomarkers from people at much younger age, that we're not starting 50s, 60s, that we're doing this in 30s and 40s, and know where you are and what your risk is, and then you can have a tailored approach, because it's going to differ throughout your lifespan, and I think we've touched on that.
So I think we're going to have a globally scalable AD screening tool. I think it's going to be blood and digital combined. I think on the digital front, it will be age, language, culture, education agnostic, so that we can accurately pinpoint someone's risks at whatever age, right? And bring in this sort of era of a brain check, so that we're really thinking about precision brain health rather than precision medicine.
So, Henrik, all this talk of biomarkers, I know one thing, you're not sleeping between now and January 1st.
It will be tough. We'll do it together.
That's right.
I agree with everything what has been said, actually. But I also would like to mention that I think if we talk about now 5 years, then we still need to care for the people who still progress in the disease. So that's also very important.
Yes.
Yeah, but I agree with everything said here.
It's good. Howard, last word.
Yeah, I think ultimately clinical care will change dramatically, because there'll be a blood test. There already are blood tests, but we really haven't seen the implementation yet of the blood tests in clinical practice, but it's happening now. So in the past, the patient would go to the doctor and they'd say, well, you have a memory problem, go home, it's probably Alzheimer's. There's nothing we can do about it. Now we can get a real diagnosis based on a blood test, so that blood tests are a common part of clinical care. Doctor orders the blood test, you get the result. And we can do that now and find out if a patient has Alzheimer's disease. The other thing is, though, that we also know now that people don't just have amyloid. They have alpha-synuclein, they have these other pathologies. It's what we call multimorbid pathology. And so I think the next phase of research will be addressing these multimorbid pathologies and then characterizing the patient based on their multimorbid pathology and hopefully having more treatments. So I think clinical care of Alzheimer's disease and related dementias is going to enter the mainstream of primary care. And that's going to be a huge difference, because, I get referrals in my consulting practice all the time of people that went to the primary care doctor, and the guy still is saying, oh, you're old, you have a memory problem, there's nothing we can do, we don't really think these new drugs work, and, just abandon patients. And it's changed a lot in 40 years since I've been in practice, but it's still not there yet where we have really good clinical practice methods yet to really take care of people. And I think also the last thing I'll say is that it was modeled a number of years ago that if we could delay the onset of Alzheimer's and related dementias by 5 years, we would reduce the number of cases by 50%, because the people would die before they became demented. And that's an amazing statistic, and I think that's doable. Perhaps by 2030, I don't know, but that's the world we're looking at as we get into more preventative measures and implementation of those measures for the purpose of brain health, that the number of cases will go down, actually.
Nancy, how great was this, huh? How about for our good prize winners?